So welcome to this video on nucleic acids. So this is kind of the third video in our series where we're looking at organic molecules or biomolecules. The big idea here, obviously, is that we have these categories of very large molecules that we find in all living things. So, whether we're talking about a tree or a fungi or a human being, we find that these exist. And so our deep dive into these is, besides appreciating sort of their interesting feature of being found in all living things, is to understand, kind of why are they in all living things by examining their structure and their function. So clearly their presence in all living things means they're doing something very important. And we're going to see kind of how does their structure make them more efficient at that essential function. When it comes to nucleic acids, there are two big ones that you guys are going to be familiar with, even if you don't realize that they're nucleic acids. And I tried to give you a hint here. And I asked, what are the two examples of nucleic acids? And you'll notice that I've underlined the letters Na. If you are thinking to yourself right now, can I think of two things I know of that end in Na? See if you could maybe get to DNA and RNA. So those first two blanks for number one. The two examples of nucleic acids we want to write are DNA and RNA. So those are the big polymers or the large molecules that we have in our nucleic acids. Now, the interesting thing about, unlike some of the other organic molecules that you've looked at so far in your carbohydrates and lipids, nucleic acids primarily exist in their polymer form. So they don't have, except for one kind of special example here, they don't really have a function as monomer other than to construct DNA or RNA. And then we'll look a little bit about what these molecules do. But these are the two big ones. And again, that Na in there actually stands for nucleic acid. The D in here, and this is trivia, this is not something that you're going to be required to know, but the D actually stands for the name of the sugar that you find in this molecule, and that is the deoxyribose sugar. So the long fancy name, if you enjoy complex science terms, for DNA is deoxyribonucleic acid. And the R in RNA actually stands for its sugar, which is ribose. So we end up with ribonucleic acid. But again, we can affectionately refer to them as DNA and RNA. In terms of their general job, we're going to kind of start broad here and then be a little more specific. So line number 2, we have nucleic acids. Their job is to store and transmit genetic information. So you have kind of this the sequence of code inside you that actually is the directions for how to make various proteins. And proteins are what are your actual physical features. So for example, when you look at a person and you notice that they have brown eyes, we know that that's a genetic trait. They inherited that from one of their parents. What we have to realize is that in order for something to be a genetic trait, it means that there's a recipe somewhere inside that person's DNA that actually tells them to make a specific protein. In this case, it's a pigment protein, so colored kind of causing protein. Inside their eyeball, their DNA has that recipe. They therefore make that protein. And so you look at them and you see brown eyes. But that information for actually creating that is in that DNA. Now, DNA is going to, and this is a preview of coming attractions, DNA is going to store that information, and RNA is going to be a little responsible for transmitting it. So I'm going to ask you guys to kind of use or branch off of number 2 and see if we can't, let's tease out those words of store and transmit. The storing of that genetic information, as I said, is actually in the DNA. DNA is the master copy of your genetic information. And as the master copy of your genetic information, our cells keep it actually protected inside this nucleus. I always think of this like if you were to go to a library and they have the original really expensive books, that are hundreds of years old, and they tend to keep those kind of, in these preserved chambers in the basement. But, if you're interested in what's in there, they're not going to, take out the book that they probably only handle with white gloves and just hand it to you to check out. But they might make a copy, a photocopy, or maybe a digital copy of some of the pages so that you can check out the information that's in there. Your cells are operating very much on kind of the same vibe here. DNA is that kind of original copy, so that preserved, essential housing of information that the cell needs. But occasionally, you need to actually take that information out of the nucleus, so kind of out of the vault where it's being kept, and you need to use it to actually make something inside your cell. This is where RNA comes in. RNA is a photocopy, so it's a disposable kind of one-time use molecule where it goes in and it sort of duplicates or copies down some of the information that's inside of our DNA so we can make something off of that genetic information. So again, for number 2, if you can make sure that next to that you've got somewhere noted in here the idea that DNA is doing the storing and RNA is actually doing that transmitting. Now, as we said, these polymers tend to exist in their large molecule form, but like all of your other organic molecules, they do follow the rule of they have a monomer, so a small molecule that you just repeat over and over again, and you link them together and you build these big, big molecules. You saw this with carbohydrates when we had the monosaccharides joining together to make the polysaccharides. And so we're going to have a similar sort of thing here going on here. For number 4, and we'll do 3 and 4 on the slide, my apologies. So the name for those monomers or building blocks are nucleotides, and the nucleotides themselves are actually fairly complex. They have 3 parts to them. When you get into the digestive system and we start talking about how the foods that we eat break down, we are going to see kind of our nucleic acids breaking all the way down to these components. So besides them being kind of interesting features in terms of the makeup and composition of your nucleic acids, you'll see them again, especially in digestion. There are 3 parts. We have the phosphate group. This is in yellow and it's circular over here on this side. This is a representation of a nucleic acid that shows you not only the elemental composition, but it also shows you the general shape of the molecules. You will occasionally see very simplified diagrams of these nucleotides, where the phosphate group that we see in yellow right here will be represented just by a circle. The sugar that acts as kind of the central point, attachment point here, is a 5-carbon sugar, so it's always going to be represented as a pentagon. And yes, that is an oxygen at the top there, so it's a carbon and oxygen ring. And then this will be often represented, as I said, just as a pentagon. And then you have your nitrogenous base. The nitrogenous bases are what actually give us our genetic code. So when we talk about the A's, T's, G's, and C's, like you'll see shorthand, like someone will say like, GGCA is the recipe to make this protein. What they're actually trying to reflect here is specifically what is the sequence of these nitrogenous bases. And these are often denoted just by like a rectangle or some shape coming off of here, but they actually are rings, which is kind of cool. So you want to make sure that you not only have these listed out for number 4, but that you also have them labeled on the diagram as well. That's kind of over on the side. It says label the image. So just don't forget to that piece as well. Number 5, I feel like you probably got this one, but if you think about the parts of a nucleic acid, of a nucleotide, we had a phosphate group. We had a sugar, which means we're talking about a carbohydrate, and you have a nitrogenous base. There are 5 elements in there. I'm sure you can probably get there with what they are. See if you can guess them. All right, so hopefully you were able to guess that we get from our sugar, we get the elements that are inside of our carbohydrates. So I get carbon, hydrogen, and oxygen. And then from my nitrogenous base, I get a big old hint that I'm going to get nitrogen. Yes, there is nitrogen in a nitrogenous base. These are also sometimes just called bases. So if you get nitrogenous base, that's just someone being a little fancy. They're telling you a little bit about the elemental composition of this. But if someone shorthands it for you and they just say the bases of DNA, they're still talking about this part. They're just telling you the shorthand version. And then the last element we get from our phosphate group, it's phosphorus. So we notice that of our 6 elements, the only poor element that gets left out of our nucleic acids is sulfur. No luck for sulfur. All right, so now we'll look at the different nitrogenous bases that exist. So just this spot here, what are the different molecules that can sit here that give us our genetic code, make me different from you and us different from trees and all that good stuff. So there's a nice constant here. Your phosphate and your 5-carbon sugar are what are often referred to as the sugar-phosphate backbone. They're what it actually gives your DNA molecule sort of its structure and shape, allow it to be that really strong, stable molecule that can kind of carry this information, so be that resource of information. But that nitrogenous base that we have right here, that is variable. That is what gives us diversity, because we can actually have different versions or different molecules. And as we swap out these kind of various nitrogenous bases, we get different proteins. So for example, based on my sequence of nitrogenous bases, I might make recipes that ultimately give me type A blood, whereas you might make a series of nitrogenous bases that based on the protein it makes, you have type B blood. And again, that variation that we see in our genetics is just due to the sequence that these bases are in our DNA. So in DNA itself, DNA is double-stranded. It has 2 sides to it. So we actually end up with what are known as base pairs. So pairs of nitrogenous bases that are going to connect together across the 2 sides of that DNA molecule. We have, and we're going to write out the full letters, the full names, just so like if you see the full name written out, you'll be like, yes, I have seen this word before. but in shorthand, we'll just go by their first letters. So the first pair we have is A and T, and the fancy name, just so we can recognize them for these one, is A stands for adenine and T stands for thymine. So these are, again, just proper names to describe these molecules right here. Notice, as a cool thing, adenine has 2 rings to it, and thymine is a nitrogenase base, just has 1. Now, what you have to realize here is that someone in these diagrams, to make it easier for you to see, has removed the sugar and the phosphate from these. But if we were drawing out a full DNA molecule, obviously we would have to put those in coming off here. So this sugar here would be attached, this nitrogen right here would attach to the sugar and then the phosphate group, and this nitrogen here would be attached to the sugar and the phosphate group. They just kind of tried to make it simple by cutting those out. The next pair that we have, again, going across the 2 sides of our DNA, are G and C. G stands for guanine and C stands for cytosine. Notice here that guanine is 2 rings and cytosine is 1 ring. So a cool pattern that we should be picking up on here is that when we have complementary pairing, so A always with T and G always with C, it's always a 2-ringed molecule with a 1-ring molecule. And that is going to be a nice feature that will keep our DNA a uniform width. Because if you think about this, if I had adenine and guanine pairing together, well, they're just bigger. But my DNA would kind of bow out a little bit here. And if I had thymine and cytosine pairing together, well, they're kind of skinny molecules, so I would have a little bit of a dip in. So to keep our DNA looking like it's kind of a wiggly line, we instead always have 2 pairing with 1, which makes it a nice even looking ladder shape. All right, RNA. It is going to have some bases that will pair. Now, I showed them here with kind of when we do transcription, which is something you'll see in protein synthesis. RNA does have some pairing when proteins are made. So they're showed as paired up here, but it's important to realize that this is something you would only see kind of as this RNA was being read by different structures like ribosomes inside of our cells. But RNA itself as a molecule only exists naturally as a single-stranded molecule. So unlike DNA, where in its actual form and structure, you would have G's and C's paired together and A's and T's paired together. This pairing that you're seeing right here is more reflective of like, what do we see at a ribosome, or what do we see when you're actually copying the DNA for pairing? So the basis, we should notice that there's a different letter here. We've swapped out thymine, which is not present in RNA, and we've replaced it with the letter U. But we've kept 3 of the same. So we still have adenine, but instead adenine is actually going to pair with a molecule called uracil. Uracil is less stable than thymine, and this is one of the reasons why RNA is kind of the disposable version of our nucleic acids. It breaks down pretty quickly, in significant part due to the fact that uracil is not as stable as thymine. And then our second one, no change here, the good old constants, guanine and cytosine, they still are going to pair together. And again, these are referred to as complementary base pairs, meaning that if I know what I have on one side, so if I tell you I have an adenine, and we know that it's going to RNA, then you know that you would get a uracil on the other side. All right. So those are your monomers. So kind of looking at their structure and their different components. The nicest one you'll have to remember in this whole unit is that the polymer, or the large molecule. So if I join a bunch of amino acids, getting into proteins. If I join a bunch of nucleotides together, I'm going to get a nucleic acid. These will sometimes, but again, a little note here, mostly in older textbooks. So like the textbooks you guys have, anything that's recently published is just going to call polymers nucleic acids. So you don't have a new word to remember. But if you happen to be digging through some 1960s bio textbooks, or maybe reading something a little bit older, then they are sometimes also referred to as polynucleotides. Again, if you decode this word, it's kind of a nice, easy one, because poly just means many and nucleotide is referring to that monomer, so the building block. So it's kind of telling you, oh, we're looking at a lot of nucleotides together. We're looking at DNA, RNA, we're looking at a big molecule. All right, what I would like you guys to do right now is pause here, and I want you to kind of take a few minutes. If you're around someone who's kind of at the same spot at you, you guys can kind of put your heads together for this one. And this is a little bit of a review. So kind of where are we so far in terms of what we've learned for nucleic acids, and then some throwback stuff on your carbs and lipids. So pause here, see what you can fill in on this chart as a little bit of a where am I at diagnostic with this stuff? Maybe do I need to do some studying? All right, so hopefully you took that as an opportunity to pause and fill this in. We're going to go through this right now, and this is a nice kind of indication for how to be successful in your biomolecules unit. So biomolecules, yes, does have a lot of language around the structures and functions of your organic molecules. And it ends up being a lot of word association, which is why studying for this often is doing stuff like flashcards or filling in charts. So here we are given in our first row that we are looking to fill in and find information. And all we're given is that the polymer for this molecule is a polysaccharide. So I'm going to look at this word, polysaccharide, and I remember that saccharide is derived from the word saccharin, which means sugar. So I'm looking at something that has many sugars in it, and I remember that that is my carbohydrates. So now what I've done here, and this is again, huge way to be successful here, is if I can associate a term I've given from this unit with the category it comes from, then all of a sudden I know the rest of the information. So if I've studied my stuff, I know, okay, well, we're talking about carbohydrates. That means my elements are carbon, hydrogen, and oxygen, and my monomer is going to be a monosaccharide, so a single sugar. I move on to the next one. So I'm given in this column that we're looking at something that has to do with 1 glycerol and 3 fatty acids. I've studied my stuff, so I know that in this land we got to be talking about our friend the lipids. So then since I've studied my lipids, I know that if I combine 1 glycerol and 3 fatty acids, I get what's known as a triglyceride. Again, keeping in mind here that these are just examples of monomers and polymers. Lipids are that catch-all wild category where anything that's hydrophobic gets thrown in there. So unlike your carbohydrates, your nucleotides, your proteins, where monomer and polymer is a little more concrete, lipids just being hydrophobic has a lot more of a wide range for what could be a building block and what we can make from them. And the elements, same thing. So we end up with carbon, hydrogen, and oxygen. Here, all I am given is that we're looking at an organic molecule that contains the elements carbon, hydrogen, nitrogen, oxygen, and phosphorus. But lucky for me, I just learned this. Those are my nucleic acids. And again, review of what we just did. So a building block for nucleic acids is your nucleotide and your polymer, a nucleic acid. So hopefully you guys were able to get that. If you struggled on this, more chart quizzes, more Quizlet, more flashcards. Because again, language. And it's weird language. I know it. That's the kind of, there's no shortcuts, no easy ways to do this. You just got to kind of put the work in on this one. All right. So the polymer. So if we look at DNA and RNA, we're going to focus in on DNA first. And when we get into kind of protein synthesis, we talk about kind of the stages of that. We'll get a lot more into RNA and we'll revisit this here. So your structure of DNA is also called the double helix, and that has to do with the fact that there's 2 sides to it and that it twists. So helical means twisted. So DNA is often referred to as a twisted ladder or a spiral staircase. In terms of the general structure, so the diagram you're seeing over here on the left, this would be like if someone untwisted or almost like ironed out that ladder. This is what you would end up with. We have the sugars and the phosphates. So if you kind of look right here, hopefully you guys can see my mouse. If you kind of look along the sides right here, we notice that we have this alternating pattern of phosphate sugar attached to the next phosphate sugar attached to the next phosphate sugar. And that is what makes up the structural, very strong part of your DNA. And that's why it's called the sugar phosphate backbone. So just like your backbone gives you your structure, your shape, this attachment of these 2 molecules interchangeably gives your DNA its structure and its shape. We then have our nitrogenous bases. So again, this is the coding, the unique portion. Depending on what sequence your A's, T's, G's, and C's are down your DNA molecule, makes you a little different from everybody else. And we notice that those sit right in the middle, attached to one another using that complementary pairing. So A is always with T, G is always with C, and they are attached by these really weak hydrogen bonds through the middle. This becomes important because we know that eventually at some point we're going to need to peel these 2 sides of DNA apart in order to copy our DNA. So the fact that they're attached by weak hydrogen bonds is actually beneficial for our cells needing to do that. If they were attached by really strong bonds, it would make them more difficult to move apart. So here we have kind of a nice review, maybe a little bit new stuff for us on the difference between DNA and RNA. This is another place I'd love to have you guys pause and just kind of see what you can fill in and then come back and kind of check your answers on here. All right, so hopefully you did that. So we have RNA, and we're given that RNA is single-stranded. So again, even though they have complementary pairings, those pairings usually only occur when there's some sort of copying or transfer of information occurring between molecules, like during protein synthesis. But in its natural state, it's a single-stranded molecule. DNA itself is a double-stranded molecule. And sometimes people like to remember DNA, even though we know D stands for the sugar, deoxyribose. The D helps them to remember that it's double. For the nitrogenous bases in here, again, you are more than welcome to go by the letters. I gave you the full names earlier, just so if someone wrote out adenine instead of giving you the letter A, you wouldn't be like, what is this magical word I've never seen before? But the letters that we have for shorthand, so we have A, T, G, and C. Again, these are the letter shorthands for those nitrogenous bases where their full names are adenine, thymine, guanine, and cytosine. And then we have AUGC. Just remember, thymine is distinct to DNA and uracil is distinct to RNA. And the two sugars, I actually gave you guys these ones on here. This is just another nice difference for us. So the reason that DNA is called deoxyribose is because do you notice how on the second prime, so the little 2' right here, that's just the designation for which carbon we're talking about. Instead of having an OH coming off, like in a ribose sugar, you only have an H. So it is deoxy. It's missing an oxygen. That's how it gets its name. Scientists are very, very creative when it comes to naming stuff. And then for location, RNA can leave the nucleus. Granted, we're talking about eukaryotic cells here, because if you're like, hey, Miss Farley, what about a bacteria? You got me. Yes, absolutely. So we wouldn't be able to use our nucleus differential between those ones. But in your eukaryotic cells, like the ones you have, we can say, oh, I had a nice circle there. We can say that RNA is stuck, can leave the nucleus, whereas DNA is stuck in the nucleus. Obviously, in a prokaryotic cell that lacks a nucleus, this will not be a good differential. So that specific identifier would not be a good difference to use between them. All right. Last one. So I told you guys that nucleic acids in their functional form almost always exist as polymers, as the big molecules. And I said there's one weird exception I'm going to talk to you about. And here is that weird exception. So the molecule ATP. So we know ATP is the energy currency of the cell. So we have this wonderful process, cellular respiration, absolutely essential to keeping us alive. Like we are functionally no longer alive once cellular respiration stops. And essentially what happens during cellular respiration is we convert the sugars we eat. So like I eat a Snickers bar, that sugar goes to my cells, but my cells can't use big sugars for my Snickers bar. So they turn it into this little tiny molecule called ATP. ATP, because you got to love the acronyms, stands for adenosine triphosphate. Adenosine, because you'll just notice that the nitrogenous base that's in this molecule is actually adenine. So there you go, adenosine. So that's just a kind of a little nod to the nitrogenous base there. But the triphosphate comes from the fact that when you look at the building blocks for DNA and RNA, they have one phosphate. Whereas this molecule, holy cow, has 3. So 1, 2, 3. And they're just kind of lined up, like almost like beads on like a Mardi Gras chain, one right next to the other. What we have here is that these bonds between your phosphate groups at the end here are actually relatively unstable. So that means what your cell will actually do is it will have a molecule. Let me see if I have any animation for this. Oh, there we go. What will actually happen inside of your cell is that it'll have a molecule called ADP. So for example, you would have this same structure. You would have your adenine, your ribosome, but adenine ADP is only going to have 2 phosphates because it's adenine diphosphate. So this molecule right here, imagine that this third phosphate isn't there, is going to go to your mitochondria, so to that power plant, that energy source of your cell. And at your mitochondria, this is where your body is literally burning through sugar. And as it's burning through that sugar, it needs to store some of that energy that it's breaking down or releasing by kind of breaking down that sugar. And so what it'll do is it'll take this ADP and it'll stick a third phosphate on there. But by sticking a third phosphate on there, man, that third phosphate is just not quite comfortable sitting on the end there. It wants to kind of pop off, and it will pretty readily. So what will happen is this kind of ATP molecule will go out to the cell, and then let's say we need to move something across the cell membrane. We need to manufacture a protein. We got to do some work inside our cell. What will happen is this ATP molecule will be picked up by some structures in our cell that need some energy so they can do some work, and they will just pop that third phosphate off. And when they pop that third phosphate off, it releases a little bit of usable energy that they can use. So again, it's that third bond. I wish I had oh my goodness, look at this, I have a pen. So there we go. It's this bond right here. That's your high energy bond between that second and third phosphate. that your cell will actually break in order to release some usable energy. And then they're reusable. So let's say this ATP molecule, that phosphate gets broken off. We use it to move some proteins across the cell. So now I'm back to having an ADP molecule. Cool. This guy just goes back to the mitochondria, gets another phosphate stuck on the end, and round and round and round it goes. And that's basically how your cells are able to kind of take energy from the food you eat and then convert it into this little kind of currency system for your cell to be able to do some work. All right, guys, you should have everything on nucleic acids. Go back, check your notes, rewatch any section of the video that you would like, and email me if you have questions.